So today's speaker is Dr. Trevor Bedford, who is a professor in the Vaccine and Infectious Disease Division at the Fred Hutchinson Cancer Center, an investigator at the Howard Hughes Medical Institute, and an affiliate professor at the University of Washington. His research program focuses on phylogenetic analysis of pathogen sequence data with the goal of making inferences that are actionable to public health. This research program spans both epidemiological and evolutionary questions and spans a number of viral systems, including seasonal and avian influenza, Ebola, Zika, MERS, dengue, mpox, and SARS-CoV-2. Welcome. We're excited for the talk.
Thank you. Cool. Okay. Thank you so much for having me. Can you hear me in the back of the room? Yes. Awesome. So yeah, I'm sorry. I tried to make it last year. Got COVID and couldn't come. Going to be talking about that today. So as we're all, I think, mostly aware, SARS-CoV-2 continues to evolve quite rapidly. So if we look at kind of like all the fuss about Delta, Alpha, and so forth, these are like little branches now. And within Omicron, there have been all of this innovation that just continues to happen month to month, day to day. And what's been particularly remarkable with SARS-CoV-2 is just how quickly these variants continue to replace existing diversity. So we can see kind of in this current world, we're getting things like JN1 that sweeps when it appears in the course of just 3 or so months across the entire world. And we also have this really fairly unique thing to SARS-CoV-2 where we get these long branches, where we think that this branch here was in some particular person for a year and a half, evolving to escape that immunity before spilling back out. But this actually happened, and we kind of know about that from Omicron, but that's actually happened 2.5 times. So it happened again with this BA.2.86 and the JN1 here. It also happened with XB, one of the progenitors of XBB. So this like chronic infection and saltational jump is another highlight here that we have that I'm not going to talk about too much. What we see in this kind of very rapid evolution is that the spike mutations, particularly in S1 domain, just increase very, very rapidly. And these saltational jumps you can see here, but even outside of that, we get a lot of evolution in spike. And so we've had, since the origin in 2020, the beginning of 2020, we've had 50 plus spike mutations. If we compare that to other circulating, evolving viruses, say like H3N2, the fast form of seasonal flu, is evolving about half as fast at a per residue basis compared to SARS-CoV-2, and that doesn't matter if you're looking at the whole period or within the Omicron period. It's evolving about 4 times as fast as H1N1, maybe 10 times as fast as the flu Bs or the fast seasonal coronaviruses. So this kind of pace of evolution is quite remarkable and not seen before or elsewhere. So in the talk, I'm going to look at how we can look at this pace of evolution, mainly thinking about frequency dynamics, where we can break up clades or lineages and see how the frequencies are playing out, and then use this to look at things like fitness flux and mutational fitness effects. And I don't know I suppose I should wait for questions at the end of the room because of this. I would normally just say interrupt me, but whatever the normal policy is. So for frequency dynamics, here there's this basic population genetic expectation that if we have an allele or a variant that's at frequency x, so I'm going to always use x for the frequency, and has a selective advantage of 1 plus x, the frequency 1 generation forward would just be this x prime, and so we can just go t generations out with this, and if we look at it, we get these kind of sigmoidal curves that takes about 500 generations for something with 2% advantage to fix. But you can do this very simple logic transform, take the log of x over 1 minus x, and you get these really nice straight lines. And then the slope of that straight line is equal to the fitness of that, or the selective advantage of that virus. And so we can see how that plays out historically with SARS-CoV-2, where we look at BA1 versus BA2 in this red line. You get this very consistent straight line on a logit scale, and that has a very similar growth rate in different countries. And this is telling us that we think this is selectively driven rather than being kind of some stochastic thing or something like this. if we go forward again to this JN1 event a little over a year ago in December 2023, we get a similar pattern where kind of across countries we get a similar near exponential growth rate, or exponential when it's low frequency. And so what we can then do to kind of do things in a bit more rigorous fashion, we can do these multinomial logistic regression models, these MLR models. These have become very popular for this situation. They work quite well. And so this actually corresponds exactly to the PopGen haploid model. So here we need the initial frequency of a variant, PI, the fitness advantage, or the fitness of that variant, FI, and we just need to normalize it. So this is exponential growth, and this is normalizing out across the different variants that are in the population. And so then we just need 2n parameters for the number of variants with, again, the initial frequency and the growth rate for each. And again, we can see that this is the same equation that we have here, effectively. And so it just happens to correspond that the statistical multi-category version of logistic regression corresponds just one-to-one with the haploid model of selection in population genetics. So Marlon Figgins in the lab has implemented these models in this EvoFR package, which is public if you want to use it. Here, the input data is just a list of locations, a list of variants. These can be whatever string dates that are observations, the number of sequences that you observe on that date. And so this should be pretty generic, not just for SARS-CoV-2. And if we then do these model fits, they generally look quite good. This is way back in 2021 across states where blue is alpha, purple is delta, and the dots are showing kind of the empirical frequencies. The lines are showing the fits to the MLR model. And we can see that just with kind of a small handful of parameters, we can get some really nice fits here. And so if we then we can pull out that fitness advantage, and we can see things like deltas 2.5 times or 2.2 times as transmissible as original Wuhan in this case. And then we can then use these models kind of operationally. So this is a screenshot from a little while ago of looking at on the live nextstrain.org site. We kind of keep these updated every day as new data comes in. And this is just showing kind of, again, that nice fit, in this case of JN1. You can see that kind of the data tends to drop off a couple weeks before, and so it's like a bit of a nowcast into a forecast that we provide. We've assessed this in this paper by Eslam Abu Samra in the lab. And so here we went back and looked in the course of 2022 and then kind of looked, if we were forecasting at this date, what would we think happens, and we can look retrospectively to see how well things did. And so this is showing data from the US. We're like, if we're looking at June 1st, this is what the data looks like of these sequence counts from different clades, and then we can project forward of how things will perform. The black line is showing the retrospective kind of what we think is the ground truth, and the colored lines are showing every 2 weeks a kind of a 30-day projection. And we see that mostly we catch the uptick and the upslope and the downslope quite well. Sometimes we get a bit of overestimate, or sometimes underestimate, but it's generally doing pretty well. If we look across other countries, you can see that in other countries that have been doing a lot of sequencing, like the UK with a whole lot of sequencing, things are quite good. In places with not as much sequencing, things are a bit more spotty. And we can kind of quantify this by looking at the mean percentage error that we're off in frequency as we look forward. So if we start with the US here, at the nowcast, we're off by about maybe 2% or something. We're about off by 5 or 6%, 30-day forecast. Whereas in places like, say, even in Vietnam, which is 30 sequences in the past 30 days, we're still getting about 10 or 15% of a forecast error. So the models seem to be working pretty well for this short-term forecasting problem. And so this is kind of how we've been using this a lot of the time, just as this applied approach. For the talk, I'm going to be focusing more on kind of these maybe more conceptual biological things that we can learn from these models. So fitness flux is this really interesting term here. This was coined by Bill Musterman and Michael Lassig in 2010. And so basically, if we have, I'm still using the frequency of a variant, I, frequency x at time t, it has a constant fitness of f of so the fitness flux is the rate of change of mean population fitness. We can just write down mean population fitness as the weighted average of things that are in the population. So we're going to just sum across the different variants, take their frequency, and multiply it by the fitness. This gives us our mean population fitness. And the fitness flux is just a change of that mean fitness over some delta t. And this is this is directly quantifiable. It's something we can measure of how quickly the population is changing. And it's kind of telling us, it's telling us about its kind of climb up these like waves of fitness landscapes. This is this old term from Fisher, where we imagine here that there's like, you can't ever reach the peak because as it's evolving to escape from human immunity and so forth, our immune systems are adapting. And so that fitness landscape is constantly changing, but it's going up as kind of as quickly as it can. We can't measure intrinsic fitness, and intrinsic fitness also like needs time to it, that we don't, the fitness of the virus depends upon the kind of moment it exists because of the immune landscape is changing in the population. But what we can get from fitness flux is basically kind of a Stairmaster analogy, that we can measure how quickly it's climbing, even we can't measure actual altitude. And so we can measure through time this rate of fitness climb and how quickly it's moving up these waves, even if we can't yet, again, measure the absolute fitness. So I'll use this term a lot of cumulative fitness flux, of how far has it climbed that Stairmaster, even if we don't know the actual elevation. So we're going to kind of operationalize this by taking 1-year windows for SARS-CoV-2. I'm using data just from the US here, just to kind of make things easy, where there's consistent data across the whole time period. Globally, it would work quite similar. And then I'm ignoring the in-play fitness variation, which exists to some degree, but not enough, I think, to kind of screw this up as what we're going to be doing here. So if we look in many of these periods, like say here, we have Delta being replaced by BA1, being replaced by BA2. Here's BA5. This is JN1. We get these kind of quite nice fits, again, to actual data. Maybe there's kind of some things are a bit off here. When sequencing is more spotty, this is more off here. But generally things, like visually at least, look quite good. So we can then like stitch these fitness estimates together across these different time periods. And we can estimate of that, we can see like that BA1 is this much more fit than Delta, BA2 is this much more fit than BA1, and kind of so on, and be able to stitch these estimates together and estimate the cumulative fitness flux across this whole period. And so now each of these violins is one of these clades. The thickness of the line is showing its frequency in the population. So Delta emerges at low frequency here in mid-2021, is killed off by BA1 here at the beginning of 2022. And that you can see kind of the actual stair-step pattern of fitness increase that we see here of these different variants. We can see this period kind of that's in 2020 and early 2021 where there's not a whole lot of action going on. This period from Delta into BA1, BA2, BA5 with this rapid change, and then still lots of evolution, but less rapid in more recent times. But overall, if we just draw a straight line across this, which isn't exactly right, but for kind of an overall effect, we get a slope of 0.58 on this log scale, and so that's a doubling in fitness every 1.2 years. So this is a large number. It basically means that if we kind of roughly to think about this, if we were to take a virus from April 2024 and drop it into the population today, it would on average leave half as many offspring, half as many secondary infections as a modern day April 2025 virus. This is very, very rapid evolution. If we do the same thing then for flu, H3N2, we can see that here, just because things turn over a little more slowly, you need to look in 2-year windows. We have this funny period in the middle of the pandemic when there's basically no flu circulating, so it becomes very difficult. So bridging this is a little challenging. The fits don't look quite as good. I'm going to argue that they're okay for our purposes. The biggest issues with fitting this to seasonal flu is we have this kind of over summer issue where like lines fit well within a season, but then like when we have these reseeding events, things can kind of bump in frequency to a fair degree. But if we then do a similar operation for seasonal flu H3N2, we get this sort of pattern where again we can draw out cumulative fitness flux. We get a slope of 0.06 or doubling of about 11.4 years. Which is kind of, in absolute terms, still a lot of evolution, but if we compare it to then to SARS-CoV-2, it's much slower. And so this gives us kind of an actual empirical measurement of how quickly these things are evolving, where like these things of comparing spike mutations to hemagglutinin mutations and so forth is a little bit apples to oranges, where this should be basically apples to apples in terms of how quickly in a population-genetic sense these two viruses are evolving. And so we get this much more rapid evolution in SARS-CoV-2 than in H3N2.
Can you say again the interpretation of...
Sorry.
Is this the microphone?
Yeah.
Can you say again the interpretation for, you said that if we dropped, say, COVID-19 nowadays, whatever is circulating back in 2020, you said something would happen. I didn't catch that.
I think this, and this is like related to this like fitness wave changing sort of thing. But if we assume that wave is constant enough over the course of 1 year, then it would mean if we took a virus from today, April 2025, and dropped it in the population in April 2024, it would leave twice as many offspring. Or if we took a April 2024 virus and dropped in the population today, it would leave half as many offspring.
So it would be more efficient?
You can think of it as like that's the pace at which immunity is being escaped, so that there's modern day viruses have a bunch of additional spike mutations that allow them to escape from immunity, and that number of mutations, if you drop those mutations in the population a year ago, would effectively double the second number of secondary infections.
Perfect. Thank you.
Yeah. So like if effectively if 20% of the population is susceptible to the April 2024 virus, 40% of the population is susceptible to the April 2025 virus. 20%, 40% is made up, but like it should be that kind of doubling of fractions susceptible. Yes.
But presumably there's some range on this where that calculation no longer makes sense, right?
Exactly.
Do you have a sense for flu and COVID, what that window is?
Yeah, no, that's really good. And like, yes, it should definitely be the case that like if we took this modern day virus and dropped it in the population alongside original Wuhan, it's not going to be whatever 2.5 log is in terms of additional fitness. And so, I wish...
10 and change, right?
What's that?
10 something.
Yes, exactly. It's not going to be equivalent to that because like much of the fact that this is better than this is due to like escaping Omicron's BA1 specific immunity. And so it doesn't, to these viruses, it shouldn't matter whether like both of these should be escaping that Wuhan immunity. And I don't have a good answer to like what that actual time scale is. With flu, like you could, the way that I would get at it is not this, it would be like look at historical sera and look at how viruses through time escape from that historical sera and kind of map out sera through time collected through different points in time versus viruses collected through different points in time. And I'd expect some kind of asymptote sort of behavior there. But but I don't exactly know the time scale of that. Other questions at this moment? Yeah, please keep interrupting. We're doing good on time. So now getting into kind of the more, still more theoretical side. We can have these models that are now, this is like purely simulation model that I worked on a where we can basically put viruses in antigenic space, and then you mutate in antigenic space, and it should be the viruses that are more different will be successful. And that causes this kind of evolution to escape from prior immunity. And then it gives you this ladder-like tree. This was designed to look like seasonal flu H3N2. And these models kind of work quite well to give you this like continual population turnover as new mutants arise and kill off what existed. And so in another paper by Richard Nair and Oskar Halachek in 2013, they had a simpler version of the kind of same concept that they're able to kind of quantify much more, where they have an idea where fitness is some point, and a mutation bumps your fitness just directly up and down. And so you have kind of this like Gaussian kernel of like how, almost like a Brownian motion of how much does fitness get mutated back and forth. And what that ends up being is you get a Gaussian distribution, a normal distribution of fitness at any given time. And that width of that distribution depends on your mutational influx. Here, this is mutation rate, and this is the mutation effect size. And then that width of that Gaussian distribution, the variance of that Gaussian distribution, determines how quickly the fitness wave translates to greater fitness. And basically, in this model, if you know the variance of fitness, you can know how quickly evolution will happen. And so there's kind of an easy process by which mutation creates variance in the population of fitness, and that variance is what drives evolution. And so we can kind of look directly empirically at this, in this case of SARS-CoV-2. And so, when we do so, we can see that this is just drawing the mean population fitness. Again, that equation that we looked at before, we can just calculate this empirically because we know the estimates of fitness for each variant. We know the frequency estimates of each variant. We can see periods where it's kind of not moving as much versus periods where it's moving up farther. And so then we can look at this and calculate just very directly these quantities that we care about from these models. So we can look at each dot is a day time step. Each day we can look at the variance in fitness. So this is like looking across the different variants that exist in the population and calculate the variance in the normal kind of statistical fashion. And we can also look each day and look at, compared to 30 days ago, how much has the mean population changed in fitness. And so we can see these then periods where during this is the delta to BA1 transition. At that moment when they're both in the population, they have 50-50, they have very different fitness. We get a very high amount of variance in the population, and that now declines as everything becomes BA1. Then we have BA1 and BA2 existing at the same time, so there's a lot of variance in the population. And you can see that corresponds to a lot of evolution. And so we get this actual quite nice correlation where slope's nearly 1, a correlation coefficient of 0.87 between the amount of variance at any given day and the amount of change in fitness, mean population fitness flux any given day. And so this was this nice empirical result that kind of corresponded to the expectation that we had here. And we can also kind of just look at more closely at what's happening over time, where we can see that in 2020 it is true that there was less fitness flux, less evolution. 2021 we have this peak. We have periods that are up here, days that are up here, much of it down at like 10 log fitness change per generation, -3, and then into 2024 where it's declined. So we have this is a bit comforting, definitely, to see that this is declined, and we get this. The other way to get at this and see this decline is that the jump to JN1 here, in terms of the number of spike mutations and the actual reduction in immunity, was quite similar to the jump here, but the actual variance in fitness was much less for JN1 than for initial Omicron BA1. So that even though spike mutations are just going at this constant rate, and spike mutations haven't slowed down at all for whatever reason, immunity, et cetera, the actual kind of fitness effect of decreasing a bit. And so then we can do the exact same thing again with H3N2. Again, things are more noisy. We get, again, a slope close to 1. Correlation coefficient's not quite as good, but I'd argue it's still doing okay. Things bump around much more. But the important thing is that flu is only going from like 0 to 3 in its dynamic range of what we get for fitness flux, whereas SARS-CoV-2 was going from 0 or close to 0 to 50, and then now more recently to 10. And so we still see this kind of very much more rapid evolution going on in SARS-CoV-2. And this, I pose this as a puzzle, but it's not at all actually a puzzle, where like this is something that we very much expect from PopGen, and it's kind of impossible not to have this. So in Fisher's fundamental theorem, we have the rate of increase in fitness at any time is equal to genetic variance in fitness at that time. So fitness flux, the mean population fitness dt, is equal to the variance in fitness. And this is just like a basic outcome of population genetics that we can't avoid. But what I really liked here, and what was helpful for me to think about, is that like if we want to slow down the rate of evolution, which seems like what we do, the easy thing, the way to think about doing that is to decrease the variance in fitness at any given moment. So we don't really have strategies that would work like this, but if you could imagine a vaccination strategy that reduces the variance in fitness should reduce the amount of evolution going on, versus a vaccination strategy that actually increased the variance in fitness would theoretically speed up the amount of evolution that's going on. And so you can imagine vaccination strategies that try to blunt things a bit more than kind of vaccinating behind the curve. For example, given that the vaccines are not really transmission reducing, we can't really do that, but like this is kind of at least a way to be thinking about this for SARS or for other organisms. Yes, and the way that we would also think about blunting that is in this kind of scheme work. This variance is determined by the mutational input. So the other way to kind of blunt things would be if we could decrease the amount of mutational input, we decrease the variance here. I'm going to pause for questions there. Yes?
So I presume that when you speak of vaccination strategies,
Yeah.
you're assuming that the vaccine has some kind of effectiveness, and then the strategy is just in the way you deploy it. Is that?
Oh, yeah. I meant here, if we made a model where, come back all the way here, that we could imagine, like, say you were vaccinating right at this moment in January 2023, and we could decide, like, we could wave a magic wand and either vaccinate with this, which I think is BA5, or we could vaccinate with this, which is BQ1 at that moment. That vaccinating with BA5 should, I think, increase the amount of variance, versus vaccinating with BQ1 would decrease the amount of variance in the population. And so yeah, I haven't gamed all of this out actually, but that you should be able to demonstrate that if you're vaccinating kind of ahead of the curve versus behind the curve in terms of your strain selection, that should actually matter for these evolutionary outcomes. But again, the issue being that like even though we expect this to matter in terms of decreasing severity and so forth, the actual effect on transmission seems to be not super strong. We've looked at this. Sarah Kobe also has done more work on this. We've looked at this as well, where for seasonal flu, it's really hard to find much of an effect of like actual like coverage changing the rate of evolution, probably because of this not strong impact on transmission.
Yeah, I guess the reason why I asked that
Yeah.
is because we have. So I wonder if it has any relationship with vaccine effectiveness.
Yeah.
Because for flu, the effectiveness is really low.
Yes.
And I would think that for SARS-CoV-2 is higher, even though,
Yeah.
as you said, we're not that good at preventing infection.
Yeah, yeah.
And so I was trying to wrap my mind around those two facts, and they seem to be a bit contradictory because if you have less mutations in flu, you would expect to be able to predict better. You're closer to the original strain.
Yeah, yeah.
But I don't know. That's why I'm
Yeah,
asking.
I think my latest knowledge, which could be wrong, of kind of in-field effectiveness is decently similar. We're talking about 30% for both SARS-CoV-2 and for H3N2. It's better for like flu B at 50% in like 100 days. And this is like now a whole other discussion, but where much of that 30% rather than like being much better is, to my best understanding, like all original antigen extend for both flu and COVID. That when I get vaccinated this year, I will like boost a bunch of stuff to Wuhan or boost a bunch of stuff to the 1980s version of flu that I got as a kid, rather than like making new good responses to the de novo responses to the new circulating strains. And so strain mismatch is somewhat of an issue, and that kind of makes this original antigen problem a bit worse. But like the more fundamental problem than strain mismatch is original antigen extend that's keeping effectiveness from being better than you'd want it to be.
Okay.
Yeah, no, thanks for the question. Oh, yes.
We have one question online from Eric Moring. I think there's little doubt that increases in fitness for SARS-CoV-2 these days are driven heavily by escaping immunity. But to what extent is it plausible that there are still, since let's say 2023, meaningful changes in fitness that relate to intrinsic transmissibility unrelated to immunity, like changes in R0? And how could this be teased apart?
Yeah, that's a good question. So I'll touch on this in the second half-ish of the talk. Traditionally, the only way to like do that is based, and the way that that's kind of empirically done is you have a new variant. We see that it's spreading rapidly. We see that it's has higher fitness, and then you just like look in the lab to see how much antibody escape there is. And if you have a lot of antibody escape, you can attribute it to immune escape. And if you don't have a lot of antibody escape, you say it's intrinsic fitness. There was like, and you can like, yeah, and like really taking the two apart is quite challenging. I'll show some things that suggest pretty strongly that it is since 2023. It's I believe almost entirely immune escape, but yeah, we'll see. Yes.
So one last. So if you might be answering this in a second. So
yeah.
Sorry. If spike mutation rate and fitness flux differences don't really correlate,
Yeah.
is there some other part of the virus? Is there some other protein that
Yes.
does?
I will get exactly.
Okay.
No, good leading. Yeah, good question. Yeah, so yeah, the next, yeah, I should have hinted more at this. The next part is mutation fitness effects. Other questions about the fitness flex? Okay, thank you for the questions. So for this, what we're going to do is we're going to take exactly the same strategy as we did before, of just taking each clade, each variant, and estimating its fitness via MLR. But for this, rather than the 30-ish, 50-ish different clades, we're going to use pango lineages. So lineages are much more kind of granular nomenclature here. There's 3,000-ish of them during this time period, but I'm only going to take lineages that have at least 1,000 sequences so we can get a good estimate of fitness for them. So that gives us 367 lineages. This is going to be very similar to this Obermeyer et al. Approach. And effectively what we can see is we get things like we can measure the fitness of XBB.1 and we can measure the fitness of XBB.1.5, and then we can look at the difference here. And we can also know which mutations exist on that branch. And so this is like effectively treating pango nomenclature almost like a phylogeny, where we've broken up these nodes, and we can look at each branch, the fitness of this node versus the fitness of this daughter node, and what mutations in spike or other proteins that happen along that branch. And so when we do so, we we get this kind of effect here, where, again, Pango is quite a granular nomenclature. So they were basically, every time there's a spike mutation, Pango folks will say this is a new lineage, which is quite useful. So there are so we're actually like most of the parent-child relationships with Pango lineages don't even have a spike mutation. If they do, they have like one or two mutations. Generally, there'll be like one or small handful of mutations outside a spike. And then if we look at the empirically estimated change in fitness, it's generally not changing much at all, sometimes changing by 0.05 or 0.1. So again, fairly kind of small granular changes to fitness here. So then if we look directly at this, this question about spike mutations, we can see that there's a strong correlation between parent-child number spike mutations and the change in log fitness. And that this is kind of like, for, again, evolutionary folks, it's kind of like a McDonald-Crichton tests. In effect, it's very hard to kind of spoof this test. That we think that on average, if you put in a spike mutation, it increases fitness. And what I'm going to be showing a lot, because you need to look at this in multiple different ways, is like how many spike mutations do we need to double fitness? We need to put in 43 of them to double fitness on average. We get a Pearson correlation of 0.32 with this relationship, and a Spearman correlation of 0.38 with this relationship. So we think that measured over this whole time period, that really it's been important for a spike mutation to mutate. And so this is why we see 50 spike mutations, is because the spike mutations are increasing fitness. If we look at the rest of the genome, we don't, just on average, we don't really see much of an effect at all, or kind of very little effect. And so we think that if we just do spike versus non-spike over the whole period from 2020 to 2025, all the action looks like it's in spike. However, we can kind of break things up a bit more. And so then I could do this at a per gene level. I was doing this to break it up into kind of the core genes that are responsible for kind of viral replication and ORF1AB. And when we do that, we see kind of, again, very little signal at all for a correlation of fitness. If we look at then spike, kind of, there are different regions in spike. So an S1, which is going to be much more exposed to antibodies in general, has this decent correlation. The N-terminal domain has not as much of a correlation, as not as much signal as the RBD. So we kind of, as expected, the receptor binding domain seems to be the main focus of this evolution, though NPD also matters. But however, I was surprised by, if we look at accessory proteins, these are things that modulate immunity, like ORF8 and matrix and so forth. And it's not matrix. What is that matrix? Okay, don't remember. If you look at these other accessory proteins that are not part of the kind of core replication machinery, these actually show a signal, which I was surprised to see. And if we can then kind of continue to use this as a bit of a microscope to look at what matters in terms of the fitness through time. So bluer dots are showing branches, Pango parent-child relationships, that are earlier in time. Red are showing later in time. So I had all of the kind of colors mixed up here, whereas here we're now separating kind of the first half of the data set, roughly January 2020 to June 2022, from the latter half of the data set, roughly July 2022 to December 2024. And so we can see that if we separate them in both early period, where we think there's going to be more host adaptation, this is flat for ORF1AB, but in the accessory proteins, we actually get quite a strong signal of mutations here that, again, I haven't looked into enough to know which particular proteins these are, which particular genes. But we get a generally nice strong signal here that attenuates quite strongly into today's more modern viruses after BA5, roughly after July 2022. And then we also get a bit of attenuation for spike, or RBD, where this is, we can double fitness by just 10 mutations early on, where we now need 42 mutations or 40 mutations to double fitness in the modern era. And so this, I would argue, is mostly what's driving that decrease that we saw earlier on in fitness flux, is that there's the same number, kind of same amount of mutations coming in, but now spike mutations are doing less to improve fitness than before. This is some amount of host adaptation and like easily escaped immunity, and now this is kind of how we're escaping immunity today. Okay, and so now, we're doing good on time. So now we can use this as a general framework to think about how we assess predictors of fitness. And there have been interesting and ways to do this. This is EVE-ESCAPE by Deborah Marks and colleagues, where they have a variational autoencoder for kind of intrinsic fitness, whether things are accessible, the similarity, and so forth, to come up with this EVE-ESCAPE score, which is going to measure the amount of escape and the fitness of that variant. And if I just plug this in, it actually doesn't seem to do any better than spike mutations. So this is looking at different time periods, January 20 to 2024. This is the whole data set. This is early data set. This is late data set. And again, we see quite similar correlation coefficients in each case. So EVE-ESCAPE doesn't I would argue any strong effect that was evident of EVE-ESCAPE being predictive is basically because spike mutations are predictive. And then I was also super excited about this approach. It's like a really cool idea in concept where you train one of these sequence language models on things, then look at the embedding space, and the embedding should have how the model represents this protein versus this protein. And you can see how far in embedding space do you move. So I coded this up in ESM2. You can use this with this, for people who are familiar with this stuff, you can use this CLS token, which basically represents the context of your entire protein sequence. It's actually surprisingly easy to plug all this in and make this happen. And you can get the embedding of one particular SARS spike protein. You compare it to the embedding of the next one to look at how different they are. So basically trying to exactly re-implement what Brian had done in his paper. And again, it seems to be no better than just spike mutations. Again, there's a lot here, but if we use a smaller data set, smaller model or a larger model, whether we fine-tune it or pre-train it, we're getting similar effects to just, the jump in embedding space is just because you're moving in spike mutations. So not meaning to throw shade on these particular papers as much as say that I'm happy with this as kind of a general tool to assess these predictors of fitness. And so what we can then do is a very simple approach is we can combine across the different proteins and come up with a model that just kind of weights things at different rates and is decently predictive of 0.5-ish in the early period, 0.4-ish in the later period of fitness. And so it seems like we're now quite, things aren't all hooked up yet. The other thing is we have nice data coming in from Jesse Bloom's lab on DMS and so forth that we'd want to integrate with. I don't want to show too much on this, but basically these seem to work quite well if you're very near to the thing that you used as reference for DMS, but they don't extrapolate well. So this data would work well if you're kind of regenerating these DMS data, experimental data quite frequently. Otherwise, they don't outperform these sorts of approaches. You need to be doing a lot of regeneration data. But what we have then is we can have a simple model that combines these as a prior and then says this new pango lineage that's just emerged and is present at low frequency, we just have 10 examples in the database, we can, I believe, do a pretty good job of predicting its fitness. And so this problem of taking the soup of things that currently exist in the population and predicting which one's fitter and how they'll play out in the next month or two, I think is, again, everything needs to be hooked up a bit better, but is kind of solved or solvable to have a decent prediction of this month or two out. Where things get very difficult and much more challenging is once we're 3 months out, the things there's new pango lineages that have mutated into existence that don't exist today, and that is a much more stochastic process for which ones happened to mutate into existence, and that is much harder, and I don't have a great solution for how to kind of do that longer term forecasting. But yeah, excited about these methods for these near-term forecasting. And with that, I'll close. I really want to thank Marlon in particular, but also Aslam and other folks in the lab for work here on this, and just note that we're hiring, and particularly interested in these sequence language models, have been playing around with them a fair amount now, and interested to kind of continue in this direction. Yeah, happy for questions.
If I understood properly, the last 5 slides you presented, you showed that the large language models perform well enough because you identify what the good features are. So basically, you show that a deep neural network or deep learning can be mapped into a linear model that's weighting the right. Did I understand that part correctly or not?
Simpler than that. So if we go to this example, yeah, this is a little easier to explain. So this is like, I'm using purely the exact numbers from this paper, and they provided a really nice updated tool that kind of gives you of viruses throughout the pandemic, what is the escape score, and that was trained to predict fitness in its own. It's not quite a sequence language model, but it's similar-ish idea where you're trying to predict fitness through this variational autoencoder. It's basically like, you can think of it as trying to compress a big alignment and then figure out what's important in that alignment in terms of epistasis and so forth. And then so this is like meant to be intrinsic fitness, and this is meant to be a proxy for antigenic escape. And mutations that are accessible, that are on the surface, will matter more, and mutations that change your biochemical charge, et cetera, will matter more. And I think this is probably largely correct, and this is probably largely not doing a lot, is my guess here. And so because of that, if you just look empirically at things that have a higher score, are things that have more mutations. And so that score is highly correlated with the number of mutations you have, and it's not giving you, like, if it was working, yes, you'd expect it to be correlated with the mutations that you have, but it also like tilt things so that things that have, like the things up here that have additional, or yeah, things that are up here and have additional mutation, additional prediction of Eve escape. Things that have additional prediction of Eve escape here might do better than they're doing over here, which actually works for yellow, but not kind of in general. And there should be, I'm still very excited about approaches with these sequence language models, et cetera, that can somehow predict fitness. But the kind of couple published examples that came out recently of Eve escape in 2023 and this really nice HEAL work in 2021, neither of those seem to work in this case. But again, I think there's probably some way to do this well. But just that these two examples didn't seem to work to do this specific prediction.
So I have like a more of a like theoretical curiosity. So as in the plots where you show the cumulative fitness flux, and also like from the Fisher's fundamental theorem. So apparently flux is like a monotonically increasing function, right? What's the intuition behind that?
Yeah.
What does it imply to have an always increasing fitness?
Yeah, I think what I'm going for. So the issue being that like what's going to be. Actually, I can go forward. Sorry. We go all the way forward here. That at any given moment we have a number of flavors of BA 1.5 dot something that are emerging, and basically only the ones that will have higher fitness than BA5, than XBB.1.5 will spread. And so things that have lower fitness won't increase in frequency. And so kind of, by necessity, the things that increase in frequency will be the things that have higher fitness than yesterday's or than present-day viruses. And so that kind of forces fitness flux to increase.
They have to beat their ancestors to survive, basically.
Exactly.
Okay.
And you could have, you could measure like basically zero fitness flux if you're kind of not finding new mutations. But I'm trying to think if you could measure. I'm trying to think if you could measure negative fitness flux. It should be theoretically possible if, like, I think it could happen. I don't know if we properly measure it, but it could happen if you have only deleterious mutations. You have a lot of them, and so the only direction the virus can go is down. And then, like, even if natural selection is trying to keep it from decreasing in fitness, if you have enough of those mutations, enough of that mutational pressure, you'll still get this decrease in fitness. But generally we should have this kind of, as long as we have accessible advantageous mutations, we should increase in fitness.
I was trying to make sure I was understanding the fitness flux part of the talk, and I was wondering when you see a high variance in fitness, is that due to just very few lineages that have very different fitness, or is it more like a distribution?
No, exactly the former. And so this is like it's a little like this variance still, you can still calculate the variance just fine if you have a categorical distribution and so forth, even though like this is assuming it's Gaussian. But that's the model is assuming it's Gaussian. But here is what we see empirically, where there's a moment where there are two very different variants in the population at a given moment. At this particular moment, I think that is what gives you this very large variance in fitness at that moment. And I think that is an interesting empirical behavior. It seems like when things have moved very quickly, it's been these big jumps. I think this is largely, but not entirely, this biology of the saltational chronic infection sort of thing going on, and that we get another decent jump here. But if we didn't have that, it would be more of this kind of more of this overlap in smaller jumps.
So building up a bit on that question, even though it might not be biologically most realistic, you were mentioning that there's effectively two ways to slow down the fitness flux, which one of them is obviously to decrease the variance, and the other would be to decrease the supply of mutations. But from what I remember, for the traveling wave models, I think you need mutation rate of at least log n, where n is your effective population size. Yeah. However, I suppose, like, if from the plot you were showing there, where it seems to like evolve in discrete jumps, that's really not going to be an option. Yeah. Because that model probably behaves more like recurrent selective switch rather than the influenza-like model where you have a relatively nice straight like line going upwards, whereas this model seems more equitable.
Yeah, I think this is the maybe response here, which might be overly simplistic, is like these things that really, like, the way to slow down mutation is to slow down the amount of saltational jumps, is to like make sure everyone has HIV treatment.
Gotcha. Yeah, that makes perfect sense, actually. Thank you.
Mark had a question online. The model makes the simplification that relative fitness is constant, at least for the duration of a strain's existence. One would then know this assumption should fail if a strain infects a large percentage of the population, or when a vaccine is rolled out with high uptake, but be okay if neither happens. Is there any signature that model fit worsens at the end of a strain's lifetime?
I think this is going to work. So there's another version of this talk. I skipped a small section that directly addresses this. Okay, so in this work, Merlin did some fancier version of the MLR model that allows fitness to change through time. And so this is like, again, can't guarantee this is going to be fully accurate, but this is our attempt to empirically measure fitness through time. These are of clades, and then we can see that there's a bit of a bend that we can see at occasions, but it's largely flat. So it seemed like, it seemed useful to have this model that can capture this, but even at the SARS clade level seems to be turning over fast enough that it doesn't hugely matter empirically. If clades exist for too long and we're not updating the nomenclature, then that will become an issue where it's the kind of empirical issue is just less due to like immunity and so forth, and more due to what we're labeling as clade, let's say BA4 or whatever it is, 22A, will mutate further and become some other thing, and then like you're just lumping those things that are genetically different into one clade, and that is actually an issue. And so it's kind of decently important to make sure that the clades or whatever the nomenclature is being drawn when there is actual change in fitness.
I think we have time for one more, please.
Okay. Thank you, Julia. So this is going back to the very first part of your talk, which is a paper that we read in Journal Club, and we had a few questions about it. Specifically, I think your baseline reference was,
Oh, the Eslam's paper, Abu Samra et al., or Katie's paper, Kistler et al.?
It was the one, yeah, exactly that one. So I think the reference that you're calculating mean absolute error from is like a pooled truth from all of these countries, right?
Oh no. So yeah. So this is good. So this is truth is just retrospective truth. So it makes it difficult for, like, say in Vietnam, when like even after we get to the end of 2022 and we can backfill all the data, it's still very sparse. And so then our guess at the true frequency through time is probably off. And so that would make this kind of conservative or whatever, that like these things are different, our forecast is different from our hindcat, from our retrospective quote ground truth, just because of noise in the ground truth.
Okay, yeah, interesting. Actually, our question was more about the fact that we understood that it was like pooled across countries, and so we were wondering, that means that you're kind of assuming that the ground truth is the same in each country, and so we were wondering, since fitness is affected in part by the mutational landscape, would it be of interest to also try to use each country's baseline reference to kind of try to disentangle intrinsic fitness from whatever is happening in the country at that time?
Yeah, no, that's really good. So a fair amount here that I won't get into too much, but that, yes, this might be also what you're thinking of. So the pooling that we did was to where if we have some place like Vietnam without a lot of data, we can help to estimate the fitness of the variants, the clades that are in Vietnam, based upon the fitness that we see in the U.S. and elsewhere. And when we do this pooled model, we improve our forecast error for Vietnam, because in the 30 days before, there's very little data for Vietnam, but there's data for the U.S., so we can kind of estimate fitness better. However, we do, I'm not going to try to show it, we do see that across different countries, there can still be estimates to be somewhat different fitness in different countries. And so we've been, Merlin has been working on models that try to learn from that and see that like these two lineages co-vary, and like they're probably antigenically similar compared to these lineages that co-vary. And so so this like differences in country or region level variant fitness, I think, can be